Welcome everybody to this third session of the meeting on theme 3 from the CIHR grant. And theme 3 is broadly focused on agent-based modeling, that is individual modeling of diseases as they go through populations. So the idea is, simulating populations of individuals on a computer, how they interact, how they go to work, how they move through subway cars, how they interact, move through hospital wards and things like that, tracking individual people, and then trying to understand how individual people and moving around might transfer disease to one another. So instead of looking at these ODE type models where you have very, very large coarse-grained view, this is a very fine-grained view of how populations interact with one another. And a very important part of that is actually once you start looking at the behavior of individuals and populations, you really need to understand how individuals transfer disease from one another. So you start to need to have a good quantitative understanding of infection mechanisms and how infection might spread in certain settings or how infections actually work within the body. To understand how individuals might transfer disease to each other. So in that sense, we've got three talks here that have to do with this notion of more fine-grained modeling of diseases. The first one has to do with understanding or the mathematical modeling of disease in the body, and that's from Jane Heffernan and James Watmow. So their talk will be on in-host modeling of SARS-CoV-2 or COVID-19 infection and immunity. So really understanding how the pathogen works when it's within a human host. Then we're going to have a talk from Ali Asghari from York University and collaborators on the agent-based modeling in healthcare settings. So going to tell us a little bit more about if you create a detailed model of a healthcare settings, perhaps a kidney dialysis ward, and you understand how patients and healthcare workers move through that area, how you can understand how disease like how COVID might spread in that setting. And then we're going to have another talk after that that has to do with, again, with the mechanisms of disease spreading or the mathematical modeling of the mechanisms of disease spreading. And it's going to be concerned with indoor humidity and how it affects COVID-19. So very, very focused in on how the setting of an indoor setting and how the conditions within an indoor setting might affect disease transmission. So they're interrelated talks, but they touch on different aspects of more fine-grained analysis of COVID. So let's get on to our first one in the interest of time. As I mentioned, this is Jane Heffernan from York University and James Watmow from UNB. I think, Jane, are you the one who's going to be doing the presenting?
Yep, I'll present.
Okay, so Jane Heffernan is a professor from York University. She's an expert on immunity and leads the Modeling Infection and Immunity Lab at York University and a co-director of the Center for Disease Modeling. She's a university research chair in the Department of Math and Stats. She's organized workshops, summer schools, mentoring programs, and developed a program in mathematical biology. And I'll mention her co-author, who is my colleague from University of New Brunswick, James Watmow. He had his PhD in mathematics from UBC at the Institute of Applied Mathematics. He's a professor at UNB right now in New Brunswick. He's a member of the Canadian Centre of Disease Modeling. His interests include modeling of ecological systems and infectious diseases, amongst other things. So please, Jane, you start your presentation.
Thank you. I'm going to speak briefly, I guess, in 10 minutes about modeling infection and immunity. And this work involves a lot of people in an in-house modeling group, incorporating James Watmow, but as well as James B, Sajad Gemi, some postdocs, Chapin, Samine, Susan, David, and then other colleagues, Morgan, Jude, Ian, Matt, Angie, and then is informed, a lot of our work is informed through discussions with David Buckridge, Nick Ogden, and Bruce Mazer. So we're interested in, I guess, first I'll say that this is incorporated into the agent-based modeling section because when you're tracking individuals and how they're moving around in a population, it's important to also understand the distribution of infection and immunity in the population as well. And so you can think about this in terms of you have an individual, this individual has an immune system, they can be exposed to an infectious disease, and depending on what status their immune system is, they can either be infected or not infected. And if they are infected, the immune system status also helps determine how severe their infection is going to be. So how high their viral load will be, what their symptoms are going to be, how sick they're going to feel. And then it also determines how transmissible that individual will be back into the population. And so we have this feedback with the population where we have our immune system determines our susceptibility. Then when we're infected, we have we're not infected or we are infected, and then we have severity of symptoms, and then also how that links to transmissibility, and we have this feedback loop going on and on and on. So when we're talking about in-host modeling, we're tracking how the immune system and the pathogen are interacting. We're also looking at how the immune system, how immunity is developed, how it protects against infection and against severe infection with respect to new challenges or new exposures to pathogens over time. We're also then trying to get an idea of what the severity of symptoms will be like, because it's going to change an individual's behavior in the population. And then we can do the same thing as opposed to looking at pathogen and immune system interactions. We can also look at how the immune system is activated given specific types of vaccines, how many doses of vaccine, at what age you're getting the vaccine. And on top of that, we can also incorporate drug therapies and how that's going to modify the immune response. And then, of course, linking to agent-based modeling. When you're thinking about an individual's immunity, then you also need to be thinking about how they make choices wandering around in the population, going to work, et cetera. And given that they have knowledge of their immunity, their choices might differ. And we also do some modeling on behavior change during epidemics as well. So we're interested in looking at the distribution of immunity in populations. And when you are looking at that, you have to first understand how infection and immunity are working in an individual. So you can think about how over time your immunity can protect you. So you have your infection, you get some immunity, and a few weeks later you're re-exposed to the infection. Either you get it or you don't. And if you do get it, you can have an inapparent reinfection. And even many years later, your immune memory or your immunity can be strong enough to be able to protect you from infection, or you could have a milder inapparent reinfection. You can think about this in terms of your immunity being on like a conveyor belt, where you have an infection and then your immunity might effectively wane over time. So either that means that your immunity inside you is decaying, or the pathogen can be evolving such that the cross-reactivity between the immunity you've gained from an infection with a particular pathogen or a vaccine that's related to a particular pathogen, how that is related to the current one that's in circulation, and then how you can have a weakened level of immunity just because the pathogen has evolved away from the immunity that you've gained. So you can have different infections over time. You can also have, here's your vaccine, you can get a vaccine, and then maybe a few years later, you get the vaccine again, and so on. But the whole aspect here really is to understand that your immunity is not static. It's not once you get infected, you have immunity. You have some level of immunity, and that level of immunity and the efficacy of that immunity will change over time, depending on the robustness of your immune system and how it can maintain your immunity in your body, as well as given the evolution of the pathogen. Okay, so when we talk about immunity, there are lots of components that are incorporated. There are lots of components of the immune system. We have innate immunity, adaptive immunity, inside adaptive immunity, you have humoral immunity and cell-mediated immunity. But what's important is that what we get are, when we're looking at data from immunology studies and infection studies, we get data that are associated with B cells and antibodies. So we get antibody measurements, and we get cytokines that are related, type 2 cytokines that are related to B cells and antibodies. We also get some measurements of sometimes of T cells, CD8 or CD4 T cells, and sometimes we also get cytokines that are related to this arm of the immune response. So when we're looking at immunology data, we have to understand where those are, or what components of the immune response those cytokines are related to, and then we can use that to inform our mathematical models by fitting mathematical models to data to understand the mechanisms of the generation of immunity, the longevity of immunity, and the protective capacity of it. Okay, so at the same time that we're modeling the immune response, we also have to be modeling, if we're considering an infection or a vaccine, the different stages that the pathogen or the vaccine can have in activating the immune system. And sometimes that also requires infecting of cells. And so here we have an infection here where we have COVID-19, SARS-CoV-2 virus going in the cell, and there are lots of different things that are happening inside the cell where the immune system is working mainly on the outside of the cell in the body. So we can also look at multi-scale models of infection and vaccination, incorporating components that are happening in the cell and components that are happening in the body outside of the cells. We have to look at that multi-scale organism that can really help us also understand what effects the immune system is having on components that are happening outside the cell and in the cell, as well as looking if we're going to start incorporating drug therapies into our in-host models, where the drug therapies are going to be working, and maybe where should the drug therapies really be working in order for us to really get rid of the pathogen in the body. Okay, so here we have just a generic diagram of the immune response. Here we have our antigen, or this could be your vaccine, okay? Our immune system notices that this is not self. We then have cells that present the not self components to the rest of the immune response through the naive CD4 T cells. We have activation here, and then we can have either our naive CD4 T cells become Th1 helper cells or Th2 helper cells, and then they go on and activate the rest of the immune response. So here we have in the Th2 arm, we have activation of B cells to memory B cells and plasma cells, where plasma cells make antibodies, which go and neutralize the pathogen. And then in the Th1 arm, we have the activation of naive CD8 T cells that can become memory cells, but they can also become effector CTL, which go and kill infected cells. And so what we really want from an outcome of infection and from vaccination is we want to have memory CD8 cells, memory CD4 cells, and memory B cells and antibodies that are going to give us that protection against infection in the future. And so when we talk about type 1 cytokines and type 2 cytokines, we do get data that are involving these different types of interleukins and interferon gamma and so on. So these are components that we need to keep track of in the data that we can link to our mathematical model. Okay. So here on the left is a flow diagram of a mathematical model that we've used to track in-host infection in individuals in a collaborative project with Mario Ostrowski of the University of Toronto. And so we have infection data from people who've had mild infection, moderate infection, and severe infection. And what we've been given data on interferon gamma, interleukin-2, and granzyme B, which is a protease that helps us measure the cytotoxic killing of infected cells. And so we get this data, and what we've been able to do is use this mathematical model on the left to look at the activity of CD4 T cells in the body and how that's related to interferon gamma, interleukin-2, and granzyme B, and then see how this activity differs in mild, moderate, and severe disease. We can, as a longer outcome, we also can look at the memory CD4 T cells and see how these individuals, given the immunity that they would gain from a mild, moderate, or severe infection, how that can be aligned with protective capacity against other variants in the future, and then try to understand what their secondary infection might look like if they do get infected. We also have another project, same postdoc that's leading it, Corusec. And here what we're doing is we're looking at the in-host infection in mild, moderate, and severe disease. So here we have lots of different cohorts of individuals from different studies in the literature over different time courses or time points in the pandemic. So looking at alpha, delta, historical strain, and so on, and during the pandemic. And then we try to determine what our infection parameters are going to be in here, and then also determine at the same time what the mutation capacity is going to be for the virus in these hosts, and then determine that if a mutant arises in a host in these infections, what's the probability that the individual will then transmit the mutant and the non-mutant virus to other individuals in the population. And so this is a study that we have in prep, and a couple manuscripts will be coming out, will be submitted soon from this study. We also, so the previous two were looking at infection. We also model the outcomes of vaccination. So on the right here is a schematic of how mRNA vaccines and replication defective virus vector DNA vaccines work. On the left here, we have a couple of flow diagrams. Here we have an adenovirus vaccine and looking at how it activates the immune response. We have data concerning interferon gamma and IL-6 that we can use to then inform what our antibody trajectories are going to look like, as well as our effector cells. Remember, antibodies neutralize the virus, and effector cells kill infected cells. We look at the accumulation of them and the longevity of antibodies and effector cells over time post-vaccination. So we also have a schematic here or flow diagram of our mRNA vaccine modeling. So we have mRNA lipid nanoparticles. Here we have vaccinated cells. They help prime the immune response for CD4 and CD8 T cells. We get data with respect to interleukin and interferon gamma. And then we can use this, this mathematical model too to look at the longevity of antibodies and look at the longevity of T cells in the body as well. So here we have our model fits to different data sets. So here is a fit to the adenovirus vaccine clinical trial data, where we're looking at the effects of one dose, which is in blue, and 2 doses in red to our interferon gamma, to our IgG response and to interferon gamma. And on the right, we have what our model fit looks like for the mRNA vaccines, where we have clinical trial data for both Pfizer and Moderna. And then what we can do when we fit these mathematical models to the data is we can first, let's look at the mRNA plots here. We can look to see what the longevity of our antibodies is going to be over time. And so here, when we're looking at both Pfizer and Moderna, we project that 212 days post-vaccine 1, where the 2 doses are given an average of 24 days apart, that 99% of our peak level of antibodies will be lost by this time. When we look at just Moderna, we have that this 99% will be lost by 238 days post-vaccine 1. And we also, for Pfizer, we have a little bit different when we're just looking at Pfizer only, but it'll be 265 days. And so what we see here is that we can use our in-host mathematical models to look at that longevity of our antibodies over time, and also then provide some outcomes, some results where we can inform when another booster dose might be wanted, so that if we're looking at, say, individuals where are getting their vaccine during the alpha stage of COVID-19, then when would they want it to get a booster dose for Delta or for Omicron? In the upper left-hand panel up here, we have outcomes for the adenovirus vaccine model. And what we've then been looking at is, well, what happens if we spread our doses further apart in time, but also what happens if we reduce the size of the second dose, which could be important going into the future when we're looking at all of these different booster doses of vaccine. Whereas instead of giving one standard dose, what happens if we separate the one standard dose into two smaller half doses that are spread apart a little bit? So these are different things that the in-host modeling project can look at. So going forward, there are lots of things that we can do. We're trying to understand infection severity by age, ethnicity, sex, and so on. So we're trying to tease apart the data, analyze the outcomes of infection by age, ethnicity, sex, and so on, where we're looking at the severity of symptoms, but also look at how high the viral load is, looking at the time span for viral load, how long an individual is infected, and then also look at how these different infection outcomes vary by different variant of concern. On the other side, trying to understand the immune system protection, quantify the protection against infection by antibodies and T cells against severe disease. But for the different variants, given how many different doses individuals have had and the time since the last dose, by age, because we know that immunity decays differently in different age groups. And then looking at different combinations of vaccines, whether individuals have had a mix of mRNA and adenovirus, or if they've had only Pfizer or maybe some Pfizer and Moderna, and so on. And then quantify the effects of waning immunity over time so that we can help identify when an optimal fourth dose would be for an individual, where we have a bunch of individuals in a population, and also considering all the different comorbidities that individuals can have. I'd just like to say that we've recently been funded by a couple new research grants from CIHR and NSREF Exploration. And these involve collaborations with the COVID Immunity Task Force, where there's lots of immunology data, the Canadian Immunization Research Network, and VIDO, that has a protein subunit vaccine that we're starting to analyze data for. And that's it. Thank you.
Thanks very much. Very much appreciated. Great talk, Jane. And I think I would probably, in the interest of time, I'll encourage everybody to post some questions in the chat. And then at the end of the session, if we've got a little time, we'll come back and circle back to those questions.